The central dogma: DNA replication, transcription, RNA translation, and protein. DNA is the replicate to produce DNA. RNA is produced by transcription. Then protein is produced by translation. Here the figure: DNA replication, transcription, then RNA translation, then protein. Today we study about the DNA replication. Long before the structure of DNA was known, scientists wondered at the ability of organisms to create faithful copies of themselves and at the ability of cells to produce many identical copies of DNA. Replication is a process in which DNA copies itself to produce identical daughter molecule of DNA. Replication is carried out with high fidelity, which is essential for the survival of the species. Synthesis of a new DNA molecule is a complex process involving a series of steps. Replication fact: DNA has to be copied before a cell divides. DNA is copied during the S or synthesis phase of interphase. New cells need identical DNA strand. So here the figure, here the S phase, G1, G2, mitosis. DNA replication takes place in the S phase. DNA polymerization requires an initiating primer which can be made from either DNA or RNA. Require an initiating primer. DNA dependent DNA polymerases cannot extend a DNA chain from nothing. They can only add nucleotide to a free hydroxyl OH. Here the figure: 5' template strand, here 3' and the nucleotide add to the 3' free 3' hydroxyl OH. Here the figure AT, free hydroxyl OH, the nucleotide pyrophosphatase, growing DNA strand, primer. Basically, template DNA and primer require a primer, then growing the DNA strand. Semi-conservative replication. Semi-conservative replication is one of each new molecule. DNA is old, template strand. One half of new molecule of DNA is new. Semi-conservative model, conservative model. Semi-conservative model basically shows one strand is old, that's called a template strand, and other strand is new, that is called complementary strand. DNA replication is semi-conservative. We know about that. Each DNA strand serves as a template for the synthesis of a new strand. producing two new DNA molecules, each of one new strand and one old strand. This is semi-conservative replication. Watson and Crick proposed the hypothesis of semi-conservative replication. Soon after the discovery of DNA structure, 1953, and the hypothesis was proved by indigenous designated experiment carried out by Matthew Michelson and Franklin Stahl in 1957. Michelson and Stahl grew E. coli cell for many generations in a medium in which the sole nitrogen source, ammonium chloride, contained 15N, the heavy isotope of nitrogen, instead of normal, the abundant light isotope 14N. The DNA isolated from this cell had a density about 1 greater than that of normal DNA. The mixture of high DNA and light DNA can be separated by centrifugation to equilibrium in a cesium chloride density gradient. So centrifugation to equilibrium in a cesium chloride density. The E. coli cells grow in the 15N medium were transferred to fresh medium containing only 14 isotope, where they were allowed to grow until the cell population had just doubled. The DNA isolated from these first generation cells formed a single strand in the cesium chloride gradient at a position indicating that the double helical DNA molecule of the daughter cell were hybrid, containing one new 14 strand and one parent and 15 strand. This result argued against conservative replication, an alternative hypothesis in which one progeny DNA molecule would consist of two newly synthesized DNA strands. The other would contain two parent strands. This